Yeah, so so there's four of us. Our team was called For Science, and so that alone was already a great learning, a great lesson, is that there were four of us and they're all in different cities. And so I think, it was in the middle of pandemic, but I think it was actually, regardless of that, I think it's a great opportunity to show that, you can do good work even not ever meeting in person. It was on Zoom. So it was me in Charleston, South Carolina, so the south of the South. There was someone in Boston, Nayara Leite, who has actually since moved to a new position. So she finished her postdoc at Harvard, and now she's at Vertex, also in Boston. So so that's her. There's also another person, Zhe Liu. She was a postdoc at UCSF. She has also moved on to a new job. Um, she's now at Novo Nordisk in China. And then finally, so she's in San Francisco, California. And there's a fourth person, Quat, who is an MD PhD student at Stanford, still is, but actually he was living part of his time in Wyoming because of computational research, single cell analysis, creating new tools for immunology. And so as you can see, we had four states represented, also a mix of levels, right? I'm an assistant professor, Nara and Jay were postdocs, and Quat was the grad students. And so also like that, so not just different places in space, but also different places in your career. And so what we focused on, so it was really was a close collaboration of four of us. We had all these Zoom calls weekly. And so we really wondered about, and now it's a topic that's become more and more relevant, is the role of beta cells in their own demise. So the idea that there must be something, maybe there's something different about beta cells themselves in type 1 diabetes. And so we used two data sets. One data set was part of the resources in the DChallenge, was this HPAP consortium work spearheaded by UPenn. And so there they have single cell data from type 1 diabetes patients and healthy controls, specifically the islets, right? So you can look at beta cells, alpha cells, and so on. And then we also used another data set from UMass. So this one was two people, one type 1 diabetic and one healthy control. And so Nayara knew the authors and the paper had been published. And so she got started on that. And then we confirmed some trends that we saw there in the larger data set to the HPAP and HPAC. At the beginning, we tried to match them by age, by stage, and also try to look on samples that had not just the islet single cell sequencing, but also cytov data on peripheral blood. But the problem there, or not the problem, but when we looked at this data, it seemed that there was not much to do in terms of having, correlations between peripheral blood and islets. So the thing there is that we kind of just focus on the single cell data. So that's not a lesson as well, is that it's not just important having a lot of data, but also what type of data and what does it mean. And so in this case, I'm not sure we saw a difference in peripheral blood, but in any case, if you go back years in literature, it's not clear whether type 1 diabetes patients have differences in their cell subsets in peripheral blood or not. So the outside is where the action is at. And so that was something that we learned as well, to have that. And so what we saw in both the small data set from UMass and the bigger data set from UPenn and the HPAC and the DChallenge resources was that beta cells has a cell stress signature, specifically in type 1 diabetes patients. And so something that I'm going to frame this at some point in my office was this really cool UMAP, right, clustering these single cell data from between healthy controls and type 1 diabetes patients. And we saw that the T1D patients, the beta cells, actually split into two clusters. And there's a cluster that is almost only, not only, but nothing biology is 100%, but almost all in type 1 diabetes patients. And so we looked at those genes and we found a lot of cell stress genes, antioxidant enzymes, and so on. And so the idea is that, well, if there's something here that beta cells in type 1 diabetes patients have that healthy controls beta cells usually don't have, can you use those as biomarkers to better diagnose type 1 diabetes? Because a big problem in the field is not just finding a cure, but also diagnosing as early as possible. And so what we did was to try to understand, or what we posited our hypothesis that can we use this, can we derive markers from this to diagnose T1D earlier? So I have a mix of autoantibodies, which usually you look for one, two, and then several, combined with beta cell stress. Because sometimes people have autoantibodies, but they don't have T1D. It could be because the beta cell stress is not there. And so that was really a big learning. So that was a science learning the teamwork across the country was personal learning, and then what type of data you use and what it actually means, another learning from kind of a data perspective. So going forward, a few things that have happened. So I've done in my lab some single cell sequencing myself. We're looking at it now in some regulatory T cells. It wouldn't be me if there are no regulatory T cells involved. And so we're doing that. And so, the work focuses on engineering regulatory T cells with chimeric antigen receptors, which can have different signaling domains. And so this was a data. So we searched the data both for new targets for these CARs and also for signaling domains that would help Tregs thrive in this inflamed stress cell environment. So in terms of targets, I'm not sure I was too lucky. Like, we parsed through the data, and then what we want. We want a cell surface marker that is in T1D beta cells, but not in healthy cells. So you could imagine driving your CAR Treg directly or uniquely to the T1D beta cell. So I'm not sure I found there was this group of genes that was surprisingly upregulated in T1D beta cells versus controls from the reg family, REG. So there's some work there in reg actually making reg proteins being made by acinar cells and being involved in pancreas regeneration. So I thought it was interesting that reg was popping out, being expressed by the beta cells themselves in these data sets that Nayar and I and Quat and Je looked at from HPAC. And so there was some potential insight. I don't have a strong follow-up on it yet, but it was something that popped up in the data sets. Again, looking at genes that are in T1D beta cells, not in healthy cells, again, with the idea to target these cells specifically for protection. I would say that that's a very honest assessment of where we are right now with this data and hypothesis.
That's great. Does anyone have any questions for the team for science? Yes, Marianne?
I think your work is very fascinating, and we just had a webinar in DKNet by Rafael Drigo. I don't know if you know his work on long lived cells, and it just reminded me of something. I don't know if this has anything to do with what you're looking at, but an old mentor of mine, I come from neuroscience, once told me that neurons are also post-mitotic and long-lived, and he said that cancer cells are looking for a reason, right, to grow and to live, and neurons are looking for a reason not to die. And I thought that was a very profound statement because, right, these long-lived cells do have apoptotic pathways in them that can sort of remove them, themselves from it. So I don't know if that's at all relevant to your work, but I just thought it was very interesting, this juxtaposition of our last webinar. And then what you're saying is we're looking, do these beta cells participate in their own demise?
Yeah, I think so. I think beta cells are under such tremendous stress, right? And there are people that say this way more than me. So there's Nayar herself and Desi Isaac, who gave recently a Meet the Expert at the Sugar Science. And so there's something that, I think these cells are always on the brink of dying as they put to the neurons. And so I think in healthy people, there's always a balance between these two states, but maybe in T1D they get locked. So I guess with single cell, get a snapshot. So what is it that there's a balance where normal people, you see more of the healthy balance, and in T1D it's locked in the stress balance? Or is it that there's a terminal progression where the beta cells have evolved, they have turned from healthy to stress, and they're just locked in there? So that I actually don't know, something else to look at. I think the mice might be a place to start. There's some work from Raghu Mirmur as well, who also gave a talk at this recently. So yeah, there's lots of excellent work going out there.
Yeah, I would also say that if we want to talk about thinking about why these cells could potentially be stressed. We just had a really great talk from Jack Verosco from University of Texas, Austin, where he's looking at the actual size of pancreas in those who have biomarkers that progress to type 1 diabetes. And it looks as if they start off with a smaller islet mass or pancreatic mass in general. So okay, if you have less beta cells, maybe they have to just work harder and they're already stressed out. So yeah, it's really interesting. And I loved your concept. I thought the way that you guys operated across borders was just really exemplary. So thank you for putting that all together. Great team for science. And thanks for sharing your your learnings. How about if we go to La Jolla, to Ferhat and Joaquin and company. You guys, you know, came in at the very top of the heap there, and love to hear what you have learned since then.
Yeah, well, good morning for those of you on the West Coast and still in morning time, afternoon for those that are already at noon. Let me just bring up the presentation. So we just put out together a few slides that I think really cover what we did and what we've been doing since the challenge. So just want to say, yeah, so good morning again, and afternoon. I'm from, my name is Joaquin. I'm from La Jolla Institute of Immunology here with the iLab. And I just want to give you a recap and updates on what we've been working on. So before I start, thanks again, Monica, and all the pseudoscience community for organizing the DChallenge and continuing to organize other events like today. I think it's important for us to learn from each other. There's different angles really to type 1 diabetes that I don't think about until I hear it from you guys. So that's really nice. And just for all the other organizers and judges. So thank you.
That's very nice of you. Jeez, got to get a better picture on there.
Yeah, I had to search this picture a little bit. I found a couple. I said, okay, this one looks like she's relaxing.
Oh, geez.
Yeah.
Okay, thanks for that. Okay, so yeah, so what went down?
Yeah, so I'll just do. Oh, what's that?
I think someone needs to mute Anissa.
Okay.
Anissa, I'm gonna mute her. Okay, go ahead.
Okay, so I just like to go over a recap and just a few of the lessons we learned. And so here in the iLab, we're really experts and we really are interested in the 3D genome. And so in order to study the 3D genome, we really have to understand two different fields. The first is genetics, right? So genetics, we're trying to study what is the DNA sequence, what are the variants that are happening within the sequence, and the different variants that we can see are things like single nucleotide polymorphisms, copy number alterations, structural variants, and there's many more, but these cover the basics. And once you have this information, you want to layer on top epigenetics. This will really tell you what are some of the mechanisms acting near, around, with these genetic variants. And so, like I said earlier, 3D conformation is what we are really interested in. And here you can see, for an example, an enhancer is connected to a promoter. And there's other types of epigenetics that you can see as well, like histone modifications and many, many more. So what is actually the interplay that's happening between these two, between genetics and epigenetics, that eventually contribute to type 1 diabetes? And so, as we all know, type 1 diabetes is a very complex disease. And in order to try to understand it, I think it's important to try to look for some of the most important studies that have been done, the most powerful tools. And so we started by looking at genome-wide association studies. These will associate the genetic variants we were talking about earlier with disease. We then wanted to include expression quantitative trait information, which will associate these same variants with gene expression levels of nearby genes, usually those that are like within plus or minus 1 MB. And lastly, we wanted to add chromatin conformation in the form of loops. This can actually create a bridge between our diseased variant and our target genes. And so if we can do this successfully, we should be able to find some really interesting and important target genes. So in order to start studying this, like I said, we wanted to find some important data sets. And I think for the contestants, I had actually started this presentation saying something like, you know, I hope that this presentation could really help the contestants. So I'm glad that that is actually the case, that it's being recorded. And so just for the contestants, I think it's really important to let them know that there's lots of databases out there. And one of the ones that we relied on a lot is this GWAS catalog. That's a database of several genome-wide association studies with many different traits. And we were able to locate one of the largest type 1 diabetes GWAS studies. And we actually used this one for the DChallenge. We then looked in literature for immune cell eQTLs. and we found this paper that had standardized two major databases known as the Blueprints and the DICE. And then lastly, we here at LGI had published a data set of HCK27 acceleration high chip data. This is actually Hi-C-like data that can extract interactions between two active regions and includes many different immune cell types. So we started with this. And so initially you have these three different data sets and you can think, okay, well, I can just, intersect them all together using their coordinates. But I don't think that's as interesting as trying to find some more interesting ways that the community has built. And so we actually use what is known as colocalization. This can actually take the summary statistics from both of these data sets, and then it'll tell you if a given SNP is important in a joint fashion, not separately, not in an intersect way, but in a joint way, is a SNP associated with type 1 diabetes and gene expression levels. And so we were able to get a whole catalog of these. And then once we did that, we then intersected our HCK27 acceleration high chip data. And so what you can see now is that we have this important SNP that we found is statistically linked to a gene. And now we also have a physical link to this. So now that we've gathered all of these, we said, okay, well, let's look at the genes that are coming up. And so one of the most interesting ones that we found was this link RNA that you can see here. We have different cell types, and it really is expressed in different levels across some cell types. And I didn't mention it previously, but here at LGI, we're known as the La Jolla Institute of Immunology because we really focus on immunology. And so we really focus on the immunology angle to type 1 diabetes. You can see that this link RNA is really expressed differently across CD8 up to monocytes. And so it's very interesting gene in terms of its transcriptional variation. We then visualized the loops or these SGLs. And what we were able to find is that this long non-coding RNA sits within the gene body of this loop in 3 different cell types. And in addition, if you look downstream, you can find this disease variant. But how do we know if this disease variant is important? Well, for the contestants as well, there's lots of databases, and one of the important databases that we found was known as Jasper. Jasper has a whole data sets of transcription factor binding sites, and we found that this SNP is actually hitting right at this conserved C nucleotide and converts it into a T, or the genetic variation would result in a T. So it's really hitting something very important. And with that, we were able to find a novel target gene for type 1 diabetes. And so we're really excited with these results. And this was the conclusion of our DChallenge project, was that we found a really important framework. We found an important gene, and we learned through the process that type 1 diabetes is a complex disease that you really have to try to find different angles to study it, whether that be through the immune cells or the beta cells, using genetics, epigenetics, or many other types of assays. And ultimately for us, we thought it was very important to use unbiased, integrative, and data-driven approaches to find novel target genes. So with that, I just want to move on to some of our updates. Since the DChallenge, we've really worked hard to try to expand our data sets, our strategies that we use, and I'll just give a summary of these things. So like I said earlier, and this should be really exciting for the contestants, I think, the GWAS catalog, we went back to it and we were able to find 3 additional type 1 diabetes GWAS studies that we wanted to include, and that seemed really important. In addition, there's a database known as the EQTL catalog that has been working really hard to gather as many of the EQTL studies out there, standardize them and harmonize them. And so before, we probably only had about 14 different EQTL summary statistics. Now we have 58. So it's really nice. It's really nice to be able to work with this larger data set. And for HCK27 acceleration high chip, we've kept the same samples. So in addition to finding these other databases, these other data sets, we also increased our strategies to link SNPs to genes. So in the first part, you saw that we used GWAS and EQTL colocalization with loop intersects. but now we are also including FindMap GWAS, which can actually look at the GWAS summary statistics and try to find out what's the causal variance or the most informative variance. And then we can finally intersect with our loops. And we also include this third strategy where we're using PyQTLs. This is a method of really prioritizing eQTLs using 3D conformation information. And so we really believe that using one method is not enough. It's really important. Not one of them is conclusive. And a recent study has shown that if you don't use different strategies, you might miss things. So it's been really important for us to, after the DChallenge, to really incorporate other strategies. So here's just a brief summary of the new target genes that we have. So you can see the three different methods. They actually don't, the genes don't overlap too much, which again indicates to us that it's really important to use different strategies. And then, so we took a union of these genes and then filtered for the protein coding genes, and we found 109. And so we wanted to actually find out, what are these protein coding genes? Are they actually studied a lot? Are they not studied? Because if they're studied a lot, then we we can't really contribute as much. But if we find some understudied genes, then we can really say, hey, why as a community haven't we studied these genes yet? So I worked on getting a consensus gene set from some of the databases that we can find publicly available. And really, again, for the contestants, you can see there's lots of databases that are ready for us to just put them together. So, so now we took this consensus gene set, intersected it with the protein coding genes. Any of those that intersected, we would say they're highly studied. Any of those that weren't, we would consider understudied. So, oh, sorry, it's not okay. So in total, we found that 26 of these were considered highly studied and 83 were understudied. We did a Guan enrichment analysis with both of them, and we found that type 1 diabetes is coming up as a top term. In addition, for the understudied, we see that there's other autoimmune diseases coming up and inflammatory-based diseases. So it looks really, really good. And we're really excited to be working with these. So overall, I think working with the DChallenge really gave us the opportunity to do all of this work. We really come from a field of the subfield of working with 3D conformation. So we're really excited to kind of be initiated into working with type 1 diabetes, applying our skills to type 1 diabetes. And there's really lots of methodologies that we were able to explore through the DChallenge and after the G-Challenge as well. As for the monetary award, I think it really has allowed us the time to find new data sets, build better models. And I didn't mention it as a bullet here, but we are mostly a dry lab. But over time, we've been working on building that wet lab. We've had projects where it's been really important to find validation, and the fact that the DChallenge was really encouraging that was really, really interesting for us, because we are moving in that direction already. And we hope that as soon as we finalize, we're close to finalizing. You saw that our target genes, once we get those target genes and maybe a publication out, we can start to try to do some tests with Jurkat cells and these kinds of things. And lastly, just on a personal note, I am working on my PhD thesis, so this work has actually helped me on that. So it's been fun. It's been a challenge, but also contributing to the PhD. So that's 2 birds with 1 stone, and that's pretty great. So with that, I'd just like to leave you with this slide with all the different data sources, tools that we used to be able to do this work. You saw some of them across the slides as well. I didn't include them here, but you saw some of them. There's really a lot that the contestants really that I encourage you to work on to try to connect together. There's lots of important data sets out there. And so, yeah, thank you guys for listening. And I hope that our experience can help the next cohort of people who try this out. So with that, I'd just like to thank my lab and the sugar science community one more time. So thank you guys. Yeah, and if you have any questions, let me know. Yeah.
That's great. Beautiful, recap and presentation. Thank you very much. And that is certainly a piece, your presentation is something we're going to share with the next cohort, like you said, because it really was great that you walked through your process and kind of give advice to the next group. So thank you very much, Joaquin. That's really great.
Yeah.
Yeah, and I mean, I wonder if you saw that new paper from the Open Targets, the Wellcome Sanger Institute in Cambridge by Gosia Triker. She's the PI on it, and it was really they mined a bunch of unstimulated cells from like 119 healthy volunteers, right, or patients or healthy people, and they looked at specifically at CD4 plus T cells, and they really kind of they overlapped, with genes whose expression correlates with genetic variations in immune diseases, and they pulled out a bunch that were specific for type 1 diabetes. And the when I saw there's a lot of CD4 in your data set, so it might be kind of a cool paper to take a peek at.
Oh yeah, thank you. I used the Open Targets quite a bit to look at my genes and see how important they are, but I hadn't.
Yeah, they used a lot of EQTLs to generate their data as well. So yeah, so cool. All right, thank you very much. And who's next? Florida?
Hi, can you hear me?
Yes. Or no?
Yeah. Yes.
So this was the team of Leandro Balzano and Melanie Shapiro, coming to us from the Mighty Lab of Todd Brusco, and which has been, sending out some really great papers recently. So very interested to hear what you guys have to share.
Thank you, Monica.
Yeah, I love what's going on down there.
Thanks. So what we did in the Sugar Science Challenge was assess some EQTL data from the Tiger study, which is a large cohort of human islets that were isolated, and then RNA-seq was performed, and SNPs were then associated with the RNA-seq data. And we basically mined this resource for hits that are related to type 1 diabetes. So as I mentioned, we used Tiger and PankDB. PankDB is another resource that is a single cell resource. And I'll just skip over this because we already introduced ourselves. So as we all know, type 1 diabetes is a very highly polygenic condition. And previously, type 1 diabetes risk-associated SNPs have been found to correlate with modified gene expression both in immune cells and in bulk pancreas tissue. But it wasn't really until more recently that these kind of associations could be made specifically in islet tissue through this Tiger resource, as I mentioned. And so what we did was we pulled out type 1 diabetes GWAS hits from this resource, and then performed pathway analysis to try to better understand what pathways were being dysregulated potentially by possessing these variants. And so what we hypothesized was that these risk variants might intrinsically promote beta cell death by regulating islet expression of genes that are involved in participation in the immune response. And although we haven't included what we found in this presentation, just as a brief overview, what we saw was that it appeared that expression of genes such as HLA class 2 may be modified by possessing risk variants at the HLA loci, and that this might impact beta cell participation and perhaps in their own demise by allowing beta cells to possibly present antigen, and that this might contribute to beta cell destruction in type 1 diabetes. And so the overall idea was that the knowledge that we gained from our analyses could potentially inform precision medicine strategies in the long term. And now that I've gone over a very high level overview of our analysis and our hypothesis, I'll give the presentation over to Leandro to discuss some of what we learned more specifically.
Hi, everyone. My name is Leandro Balsano. And yeah, basically, the things that we learned about from the DChallenge is that this allowed us to gain exposure to several open access data sets that we didn't know they exist. And some of them were very interesting for us. And some of them we knew, but we never dive into it enough, deeper enough to be able to know what they have and then to be able to use it. And the other very interesting thing that we rescue from this participation here is that it allowed us to connect with some other groups, and we are very happy that we still have this open channel, the Slack channel that we all try to use constantly to be able to perform and to create collaborations for a near future, and to try to delineate more and more ideas that we could have. And one of the last thing that we could consider it was very helpful for us is that we learn how to create and polish, let's say, a little bit a particular submission, a particular document to be submitted, like in this case, the JDRF and the T1D fund. Sorry. I think that aspects that can be improved in terms of the DChallenge, we took our liberty to express ourselves here. And one of the things that we could consider is that perhaps it should be interesting to extend the duration of the challenge to allow deeper dive into the resources that are available, perhaps starting sooner during the year. That could be a possibility. And other interesting thing is that the publicity perhaps could be increased so that the DChallenge could be known further and further. And also that could allow more researchers to participate. And also, I don't know if they could participate right now, but perhaps allowing the possibility for international groups to participate will be very interesting. And I think one of the things that are pretty interesting is that we could be able to pair those that are less experienced in bioinformatics with the people that really are, and perhaps you could offer some training sessions or office hours in which certain people could participate and ask questions about a particular database. Sorry, I keep continuing. So, and how participating in the DChallenge helped our research? I think three main aspects here. It provided us, as I mentioned, with knowledge of these tools that can be used to test our hypotheses. And also, they allowed us to develop some skills that have facilitated similar QTL analysis from our own data sets. So we've been able to use these databases into using our data, and also being able to develop and perform some of our own data platforms like Paintomics 3, and recently we published Paintomics 4, and by that manner we can be able, or we are able now, to integrate that information that we have in the wet lab to the bioinformatics platforms that we have, and by that manner we identified some interesting genes that and biomarkers that we need now to evaluate and validate experimentally to be able to publish it and to confirm these results. And these validations, well, we plan to perform these validations by using the money that the DChallenge gave us, but unfortunately the award hasn't helped us yet, because we couldn't be able to allocate the money here for several bureaucratic reasons, but the idea is to use the money to mainly three aspects. One of them is to try to validate these genes that we already identified, and some others, it's that we plan to buy some high performance storage space here in the Hypergator, which is a supercomputer that we have here in the UF, and also perhaps using part of the money to participate in some conferences to divulge the information that we already have and we are trying to validate right now.
Leandro, what is the price point for buying storage at Hypergate? What's the range?
Well, you can buy as much or as little as you want. We right now have important amount of space here, and actually we would like to gain more in computing abilities, like in faster computing, and that's not that much. I mean, you pay, usually they have rates for several years. You can pay for 1 year, for 2 years, and certain amount of terabytes or gigabytes in case you don't have that amount of money. So,
Well,
but
what
yeah,
would be a good, just sort of for future, what would be a good, you know, price point that you might want to target?
like an amount of
Yeah.
money
Yeah, you can think about it too and just text me after if you want, or.
yeah, okay, but yeah, for the things that we did, and for about 2 to 3 years, perhaps between $10,000 and $15,000 could be reasonable.
Okay, great.
Okay.
Thank you.
And with that, I want to thank you, everyone, and thank you, Monica, for all the effort in coordinating this group and create the foundations for this sugar science group to develop and to grow. And I hope, well, second of all, thank you to Melanie, because without her help and without her insight, this couldn't be possible. And also, I like to thank everybody, and we would like to keep participating. Let's see if we can in the near future. So thank you very much.
Yeah, thank you guys, and great feedback as well. Yeah, we're going to take those into consideration as we build out the second one. The first one actually was global, but the teams that sort of made it forward to the endpoint obviously we're based in the US, but I do think that you're, having someone as a bioinformatics expert as a touch point would be very helpful. And that was we had that as part of our strategy in the beginning, but we weren't able to pull it through. So that's something to bring to our next internal meeting. Thank you guys very much for that input. Okay, and last but not least, Michigan. Hanrui, hi.
Hi, and yeah, I also prepared some slides.
Fantastic.
I'm not sure if you guys can.
I think Florida has to get theirs out.
Oh, okay.
Or stop theirs somehow. There we go, maybe. Great.
Okay.
And now you can share yours.
Okay. Can you guys see the screen right now?
Yeah, you may want to just get it up to full screen. Yeah, perfect.
Okay. So thank you guys. So it's very exciting to hear all the previous team's presentation. And so this is our team's presentation for the follow-up. So my name is Henry Zhang from Dr. Yuan from Guan's lab, University of Michigan. And Yang is he's also here today with us. He's one of our teammates and has been helping us, has been helping me basically in during the DChallenge. My mentor, Yuan Fang, she's not able to be here because of time conflict, but she also sent a greeting to me. So thank you guys for organizing the challenge, and it's great experience. So I'll just start our presentation. So our project title is to achieve early diagnosis and risk prediction of T1D by machine learning. Let's first revisit the dataset we use. So actually the most data set we use for demonstration is the public transcriptomics provided by DChallenge. So all those are from GEO, and we also did the visualization of all the T1D health control used in all of this data set. And the other data set we try to use as the training data for the machine learning models were from public, from ND networks. Can be found from ND networks, which are from including human biological data interchange, some from UK Biobank, and this also some from our in-house Michigan Genomic Institute. Those are clinical GWAS information or even demographic information from T1D patients and healthy controls. And those has helped us a lot in building the machine learning models, because we know like machine learning models mostly based on the data sets. And also we used some of the SPP data sets DChallenge has provided us trying to do the optimization in feature engineering sections. So our hypothesis was actually based on the current situation challenging T1D clinical monitoring, because as we all know that there are already a lot of genomic, well-known genomic biomarkers of T1D, which can be used as early as before the baby is born. But the whole process of T1D is very, very hard to evaluate, especially for the activation of the immune activation. Even like we know, like before the symptomatic T1D has occurred, there are actually a lot of biological a molecular process has occurred before that, but it's still hard to find like when exactly this kind of process has taken place, because T1D is not a reversible process. So the early we could identify this kind of process, the better. So this kind of situation has been identified in many of the clinical data sets, including I put one of the example baby data here. It's of course one of the most classical or well-studied clinical data sets of T1D. So although most of the babies has presented antibody at one point, but there is some still huge number of baby who has been pre-diagnosed with type 1 diabetes biomarkers like genetic traits, but they're not antibody present at that time. But we know like some of the babies will still progress to T1D at one point. So this kind of process present huge challenge of T1D monitoring. So that's when we realized that the T1D clinical data set is huge censored data set. So that's when we decided to kick in with our computational algorithms. So that's actually one of the expertise in our lab. So our lab previously has developed one of the popular or like advanced survival rank model called Guan. This like actually a classical model in this kind of situation is Cox model or a random survive forest model. But the Guan, the advantage over those classical survival model is it used the complete ranking probability analysis instead of a Cox model, which only covers the uncensored data sets in the clinical the patient data. So, so due to this kind of reason, Guan has performed much better in most of the situation when evaluating the survival risk of patients in clinics. It shows the results here. And also like Guan has another advantage because it can be integrated with all kinds of, or actually any kinds of machine learning models, which the traditional Cox model cannot do. So that's how that we can integrate any kind of clinical data with Guan to predict the survival risk of clinical patients. So some of the application has been successfully carried out on different kinds of disease, such as breast cancer, multiple myeloma, by applying time series data or even image from the patients. So that's when we decided that Guan could be a perfect model for T1D risk evaluation here. So that's basically our inspiration. So our whole hypothesis is based on Guan. So first of all, we can evaluate the clinical T1D risk from the public clinical data set using Guan. So using Guan, we can have a complete risk evaluation of all the T1D patients, whether they have symptom or not. And so then we can like integrate Guan with some state-of-the-art machine learning model. So we choose like GBM here because it's a fast model. It's a fast model which is suitable for large data sets such as clinical data set, and you can integrate it with a large kind of feature set, such as you can use antibody tests there. You can use genetic traits here, such as HLA, and you can also use transcriptomic profiles from patients to monitor their real-time situation. We also propose to use SPB networks to tailor the expression profiles better to suit the T1D phenotypes. And we use machine learning interpretation method to evaluate if the or to visualize the decision-making process of machine learning model. So basically, you can use sharp analysis here, which is a very popular AI interpretation method often used in data science competition. We believe in using this kind of method, we can visualize the top factors or even biomarkers of T1D progression. So this is basically our hypothesis here. During the 2 months of T1D, actually we learned a lot. First of all, we actually faced a lot of challenges. We're actually outside of T1D. Before this challenge, I never involved in any study of T1D here. So first of all, a lot of things to learn, but we also appreciate the Sugar Science and DChallenge organizers. They sent a lot of state-of-the-art research during this process through Slacks, and also provide numerous resources for us to explore. So first of all, we spend a lot of time in learning new stuff. And also, wait, did I skip one of the slides?
That's okay. Even though you may have taken a long time to do your due diligence about T1D, you did a pretty good job. You know, I mean, you learned, you came right up to speed and were able to use your paradigm to really dig into it. So, I mean, congratulations.
Thank you. So one of the challenges, we have to like find the spot of T1D that we could fit in, because our lab's expertise is computational model, but still we need to know something we can work on in this field. We need to know what kind of problem is best suited for what kind of algorithms. Yeah. So we actually did a lot of research. So this kind of inspiration actually came kind of like during the middle of this process, because we did a lot of background research at first. So it is kind of challenging for us, but we're still like made it through. We're glad we still made it through. But still like have to appreciate Yang here, because he was kind of working with us, but we didn't know what to do at first. So we didn't do a perfect teamwork at first. So that's what we could do differently, I think, is if we could do it like do it all over again, we think we could do a much better coordination within a team. Yeah, so we also learned so basically I mixed the two slides, I'm sorry. But we also learned a lot of things like the DChallenge provide us numerous resources. So we appreciate the DChallenge provide us the data portal, which is which they provide us the a perfect platform for us to explore the numerous data set, because we know data set is important for bioinformatics research. It's basically the more public data set, the better. So we know this kind of availability for us and to do bioinformatics research or build our model on T1D. So that is important. So and actually it's very interesting after this challenge. We are still actually discussing with my mentor about the carry out of a project, but also we got reached out by a lot of other institutions or T1D in-field researchers. So we've been reached out by Caswell, T1D Institute from UMich. So they have we have meetings together, and we're in discussion of collaboration with other on T1D research within UMich. So we believe this kind of process will really accelerate the collaborations with us to T1D experts in this field.
That's fantastic. Yeah, I had heard that Michigan was, the Caswell was working with, you know, the eye surgeon, you know, group where they're trying to, they developed a whole AI ML way of looking at that disease state, and that Caswell was going to collaborate with them more robustly. And what you're doing seems like a perfect fit. So that's fantastic to hear.
Thank you. So also about the monetary award, first of all, we appreciate that, because during the COVID process, we know like there's a shortage of global computation equipment such as chip graphic cards. So appreciate that will help us to get that equipment for our computational experiments. And also I would see this kind of monetary award more as an honor than it's materialized award, because it will inspire young researchers like me, who was only second-year student, PhD student at that time, and Yang, he was only a master's student at that time. He's now be admitted. No, he's only an undergrad student at that time. Now he's being admitted to graduate school. So I think it's a very nice start for both of us to being awarded this kind of international award, and appreciate that. And I think it really will inspire us to step deeper into this field.
That's fantastic.
Thank you. And I thank everybody, especially Monica, in organizing this challenge for us in this meeting for us. And I was also be appreciated to my mentor and University of Michigan, my supportive carry out of the challenge. And I wonder if you guys have any questions.
That's excellent. Does anyone have any questions from our group? Looks like Marianne has to.
I was just putting a comment in there.
Oh yeah.
I think
Yeah.
I think all of your presentations were fantastic, and it was exactly the types of things, collaboration, right? New avenues, bringing new people in that we really wanted to inspire with the DChallenge. So you've inspired me, at least, and us to, I think, do even better next time and expand it. And we'll think about how to build upon the learnings that we have from you, because you were the pioneers, but it certainly sounds like the DChallenge is worth keeping. Yes.
Yeah. Fantastic.
Yeah. I just wanted to ask a question. Like you mentioned, you use chat values, I think. How helpful have you found them to, find important features? And was there like one feature that you thought was more important for your model? Over there?
Yeah, but actually we haven't carried out the analysis on T1D yet, but I have been like using this in a lot of other experiments. We have done other like malaria top genes and related to drug resistance, or like some top genes for cancer, which is one of the projects I'm doing right now. So I think, do you want examples, or do you want to because there are other different kinds of visualization process of like tree-based model. So some of them are based on weights, some of them based on sharp analysis. But the advantage of sharp analysis is it is kind of universal. So it cannot only be used for random forest or certain kind of models. It can use by a wide categories of models. So because the nature of sharp value, it's addictive. So it can also you can carry out a lot of manipulation. I'm not saying manipulation, but you can carry out a lot of downstream analysis on the sharp value, because it's a simple. It has a simple addictive characteristic of its value. So if you're looking for not only one single gene, if you're looking for a group of genes, you can basically add them together to add the absolute values together to know the importance of whole groups of genes. Because
it's nice. Wow. Okay.
Yeah, I would encourage you guys to go back and forth on Slack, continue the conversation. And I think you have some symbiosis here, and you can kind of learn from each other even going forward. So, and I hope that will happen. I do want to ask you just as something to think about, and if you want to just drop or say something now, that's great. But if you want to drop it in the Slack, that's also great after some thought. Here's the question. What are the top databases right now in autoimmunity? If you could have unfettered access to any database, public or private, what would it be? And it you would have unfettered access to any public or private database, and you'd actually have somebody who would, do the metadata on it for you. What would you choose? Think about it. I mean, if you want to throw a few out now and then put some on Slack, that would be great. We're developing sort of a set of targets, actually.
I think for us, we'll write it because, I think
you have a few. I saw on the slides. So the invitation is open to put it up there on Slack, but from that, we want to just maybe we'll tap into or highlight some of the ones that are already part of the DKNet access suite, and if there's ones that aren't there, we're gonna connect to them. So anyway,
in general,
sorry,
there's the great resource, the UCSC Genome Browser.
Yeah,
it's already available, but it has so many tracks And actually during my PhD, I used it a lot to even make figures, because you can add your own track and add your own data. Let's say you do DNase hypersensitivity, you do chromatin mapping, anything, you can make a track on your own to there. So it's already available, but there's more and more layers to it, and it has EQTL. So in our case, we want to care about not just what genes are SNPs, but also in which tissue they like to be expressed. And so something that's a gigantic resource.
And that's UCSF genome browser?
UCSC, Santa Cruz.
Oh yeah.
Santa Cruz Genome Browser.
Okay. Yeah. Now, and anybody can go and can access that, correct?
Yes.
Yeah. The Santa Cruz team has some deep knowledge in that space.
Yeah.
Sure.
Yeah.
That's cool. I mean, if you wouldn't mind, just put it on the Slack team, on the Slack feed, and just so we can have sort of a list in writing, would be great.
Yeah, because I think like our lab, we mostly focus on publicly available data. So I don't know if we really thought, at least I personally haven't like tried to work with anything that's, you know, you have to ask permission. So maybe you know some databases that are kind of have that that kind of permission level. But we'll see. We'll look around.
Yeah, just think about it. And you know, even if it was a private data set, or even, you know, a company data set, like, would you love to be able to get behind the firewall at Immune AI or something like that, right? And just think about that. And as we sort of try to maybe extend and develop public-private partnerships, that'll be out there. So just sort of a wish list. We'd love to hear from you. And I thank everyone again for participating in this call and taking the time out of your busy schedules to really share all these great, valuable lessons and feedback. And so appreciative of everything you are all doing in the T1D space. It's phenomenal. And it gives me great hope just to see how much you have done and accomplished in such a very short time. So I hope you guys have such a great rest of the day, and keep your eye on social media. There'll be more information coming out on DChallenge 2022 very shortly.